Welcome to another episode of the Agronomy Highlights podcast. I'm your host, Ryan Spelman. I'm joined here with my lovely co-host, Justin Brackenridge. Justin, how are you doing today?
I'm doing really good, really good. Season 4 has been kind of off and running now. We're kind of hitting our stride with this, and looking forward to more podcasts talking about ag innovations.
That's right. We have a continuation today of our first episode in the season. And this episode, again, it's an ag innovation episode. And so we're going to focus on the work being done in the cover crop cocktails field plot. We have two more researchers that are going to come on and join us. Now, if you want to hear a little bit more about that cover crop cocktails field plot, I urge you to go back to episode 1 and just at least listen to the intro so that you can get an idea of what this plot looks like. But essentially, it's a plot where they have all different types of cover crops that they're growing in rotation with corn, soy, and wheat, and studying different interactions in that field. So with that, Justin, you want to introduce our first two guests here?
Yeah, so if you remember on the first one, if you listen to it or if you remember, we met with Jared and Jennifer who were studying plant interactions and soil microbe interactions. Today we've got Theo and Allie who are working in these same plots. Theo is working with the mycorrhizae, the fungal side of interactions within these plots, where Allie is working with the the perennial nature to this, right? So it's taking a little bit different direction, but what they're looking at is how they can establish some perennial plots in here and how that's affecting weed seeds below ground and weed germination above ground. So kind of an interesting take about getting some perennial forages and some perennials back into our annual systems.
That's right. Yeah, I think we have some unique interactions to talk about today, and again, a little bit different than some of the agronomic stuff we usually do. But I think we can all learn something very interesting here today. So, with that, let's jump into it. So, Justin, next on we have Theo Newbold. Theo is a PhD student in Gretchen Koldau's lab, Dr. Gretchen Koldau. They are a fungal mycology lab. Actually, one of the labs that I worked in when I was a graduate student.
Yes,
So
as soon as you
there,
said fungus, I knew you had to be involved in here somewhere.
that's right. This is how us fungal fanatics work our way into the agronomic world. So I have a fungal fanatic on today. Theo, I'll let you introduce yourself. So if you could just introduce yourself and talk a little bit about what your lab does.
Yeah, so thank you so much, Ryan and Justin. So again, my name is Theo. I'm a fifth year PhD candidate in the plant pathology department at Penn State. I'm co-advised, so one of those people is Gretchen Koldau, who is myco kind of toxicology, mycology, and then my other advisor is Sharifa Crandall, who is soil-borne disease pathology.
Can you talk a little bit about some of your work in the cover crop cocktails plot and what your focus is in the fungal communities?
Yeah, so I think starting kind of high level, most of my research, whether it's with the cover crop cocktails folks or externally, is focused on this group of fungi called foliar fungal endophytes. And so foliar, we're thinking about leaf tissue, and then endophyte, which is often a loaded word for a lot of different people, but I come from the sort of plant pathology slash ecology space. And so what that word really means is that these are organisms, in my case, fungi, that are colonizing plant tissues asymptomatically, so without disease symptoms, for at least part of their lifetime. And so what this can mean is that these organisms are potentially beneficial. They could just be sort of hanging out. We really don't know what they're doing, but this also includes pathogens, which we are finding out more and more now can live in plant tissues without causing disease for periods of time. Within the context of the cover crop cocktails, my work has largely been focused on thinking about how different cover crop monocultures are shaping these foliar fungal endophyte communities. The context really being that there was some work done in 2022 that looked at disease symptoms. I believe it was associated with gibberella ear rot, stalk rot, and some seedling blight. And what they found, this is not associated with my lab, but or I think in collaboration with Gretchen. But what they found was that in the, I believe it was the radish plots, or maybe it was clover, there was reduced disease symptoms. And so the question came out with, well, maybe there's a cover crop legacy effect in regards to pathogens, but maybe this is also associated with this sort of plant endophytes or sort of like larger scale thinking about like microbiome context. And so my work started there. And so I've been working in the plots. I worked in 2022 and 2024. And really the broad questions of this project were, how does plant development shape these communities? And then how does management style very broadly shape these communities? And so what this looked like is we went into the cover crop site. We were looking at monoculture cover crop treatments. We had triticale, clover, forage radish, and a fallow treatment. And that was sort of our representative of organic management to try and encapsulate the sort of variety of approaches used at the site. And then we looked at conventional very broadly. So across the street over at Rock Springs, we had a couple of fields that are typically grown in sort of a maize and soy rotation to provide some funding for the research station. And so we really were comparing the sort of organic versus conventional systems very broadly. And so what this really looked like was going in and sampling kernels at planting and at harvest, and then going in and doing leaf sampling across development. So very incremental stages. So we had 2 vegetative stages. We sampled at tasseling or flowering, and then we sampled at 2 reproductive stages, and then at plant senescence, or essentially when the plant is dead, everything's dried down, and we might typically think of this as being around the time that we would harvest for grain. And it went in and characterized the total fungal communities of those leaf tissues and seed. But also, one of the questions we really had was around Fusarium, right? We know these are really important, specifically Fusarium graminearum and verticilioides are really important pathogens of cause ear rots and stalk rots. But we don't classically maybe think of them in the literature as being part of this foliar fungal endophyte group. But a lot of work out of Dr. Koldow's lab and in other literature has really identified them continuously when we do sort of more isolation-based approaches, where we would like go in and actively culture out fungus from those leaves. And so we really wanted to focus on Fusarium, thinking about them as being part of this endophyte community. And so that's kind of where my work is right now with them. In 2024, we sort of expanded on this or tried to refine it a little bit more, where we reduced the number of sampling times. We went in and rather than sort of looking at these sites as being very like realistic representations of organic and conventional, where we have maybe 2 different separate like varieties planted at each location, where there's a lot of potential kind of covariates or variation that's kind of hard to pull apart. We planted the same varieties at each site. And then we also increased our replicates for those cover crops to potentially try and pull out any potential cover crop effects.
Awesome. Well,
So I have like a
yeah, go ahead.
laundry list of questions after this, right? There's so many terms and so many things, and anyone that listens to the podcast knows that forages and grain crops are kind of more my thing, and Ryan has this like fungal fanatic, right? So I just want to ask the question, like, typically pathogen is kind of carrying this negative connotation, right? And the way you're talking about, we find these amazing pathogens, right? Like you guys are interested in finding all of the bad things that are out there so that then you know how to avoid them. Is that kind of what we're looking at?
So very broad. So I would say that my research is sort of like fundamental. We want to know who's at the party. We want to know who's in the plant. Um, for sort of long-term picture, being that this could be related to disease management or maybe mycotoxin management in the context of those Fusarium species, specifically for like our tillage, or sorry, our silage folks. So that's kind of where I'm coming
Okay.
from.
That helps. That makes it, context for me. So the other part that I want to kind of ask about is we're talking about, I think I'm not going to get my terminology right, but the leaf established colonies of these things, right? What did you call that?
I said foliar fungal endophyte, but you're
Foliar
right? They're
fungal endophytes.
yeah.
How do you get in there and you find one or three or whatever you're looking for from this foliar endophyte?
That is a beautiful question because classically, what would be done is incredibly time intensive, where we go out to the field, we collect leaf samples, we come back to the lab, we surface disinfest them or clean these leaves in some way. We cut them up, we put them on different types of media. Or like nutrient sources and try and isolate or grow out those fungi and culture them onto more media and then characterize them usually phenotypically or like what they look like. We might do some sequencing where we like isolate DNA and look at a particular region to try and classify the species. I'm coming from really much like we might think of as like the microbiome space, right? Which I know is such a buzzword, but really what it is that we're thinking about everything that's living in the leaf or everything that exists in the leaf at a period of time. And what my approach looks like is very similarly. I go out to the field, I collect a ton of samples. Thankfully, because I'm not doing a bunch of culturing, I can just collect exponentially more samples. I come back to the lab. Again, I clean those samples. I do what's called total DNA extraction, which is where I take the leaf sample, I mash it up. I use different chemical reagents to pull out essentially all of the DNA that's present. And then we do something called meta barcoding, which basically is, I know, which basically is there's this region of interest in the DNA that I know allows us, or that we know allows us to identify particular species or genera, so particular fungi. And so I go in and I essentially amplify that region in this DNA over and over and over again. And I use a special machine called a sequencer, and it spits back at me millions of sequences, just like strings of DNA, essentially, that I then go into the computer and use various programs to clean all of that up and associate it with already known regions of interest so that we can do typically genus level characterization.
So I'm not like shading any of our other guests, but like this is real science. I mean, this is like this is real business here. And I'm like
Listen, that's
listening to
what us
this.
mycologists do and plant pathologists. We do some real science over here.
I'm listening, and I'm sure you both are looking at my face like, I mean, I'm grasping at everything I can to try and keep up with this, but this is really pretty incredible stuff you're working on.
It's definitely not perfect, right? So these techniques are fairly new. There are aspects of them that can be more challenging. So like for instance, I talked about I'm looking at Fusarium as part of this overall community. Fusarium, as well as other groups of fungi, can be really challenging to use what are called like universal primers, which are just regions of interest in the DNA that we can typically identify a species with. But Fusarium are really challenging because the common sort of region that we use, which to throw another term at you, is called ITS, but that typically that region in Fusarium, we have multiple copies or tons of variation between like individual isolates within the same species, which make it very challenging to identify to the species level, which really matters if you're thinking about something like Fusarium graminearum or Fusarium verticilloides. So we have to go in and use a totally different region. And it's pretty novel because it hasn't been used very often. And there's been tons of struggles getting it to actually work for my approach. So again, it's not perfect. It sounds like, this is like really, really awesome science, and it is, but there's definitely tons of challenges.
Yeah, I think to add to what Theo's talking, just because I have some experience with these pathogens, I did my thesis work with Fusarium species, and so Fusarium graminearum causes gibberella ear and stalk rot, and verticilloides causes Fusarium ear rot and stalk rot. Vomitoxin or DON is released by gibberella, and then fumonisin and some other stuff is released by Fusarium. So one of the things and why you're probably very interested in looking at those is that we see them being a root pathogen, a stalk pathogen, an ear pathogen. And sometimes when you actually go in and see how much fungi or fusarium is in that ear, that stalk, that root, um, it doesn't always align with how much mycotoxin is being produced. So it's really tricky that we know sometimes these things are hanging around and they might not be causing disease sometimes, and we don't where classically we think of ear rots coming in when we see insect damage and then the spores inoculate and take over the ear. There's also a lot of questions on how much does it actually travel within the plant and then just become a disease. from the stalk into the ear, at different times if conditions are right. So there's a lot of questions surrounding like its actual lifestyle around the plant. So my question is specifically like considering that and knowing why you're interested in specifically this genre. Um, did you find that cover crops were structuring the community of Fusarium or other endophytes in this manner? And yeah, did you find a structuring a pattern?
That's an awesome question. So relating to results, right? So we didn't find that cover crops or a specific cover crop really had a legacy effect on these foliar fungal communities. And neither did they have this effect on the Fusarium subcommunities. And instead, what we found were that common pathogens, like those associated with like Cercospora, so like with Cercospora leaf spot or southern corn leaf blight or northern corn leaf blight, were commonly found as endophytes or these asymptomatic colonizers in these communities. And to note, what's really important, and I'll continue in a moment, but what's really important to note is that when I'm sampling leaf tissue, we're sampling green leaf tissue. There are no disease symptoms. We're not sampling leaf tissue that's been damaged by insects or other herbivores. So what we're looking at really are organisms that are these fungi that are colonizing asymptomatically. Um, and so we found that these common pathogens are essentially fairly ubiquitous across time and between cover crops. And we found that between sort of these two sort of large scale management styles, right, between organic and conventional in our representative fields, that management was not a driver of the Fusarium communities. And that instead, what we found is that really host development is shaping these communities where they're more diverse around tasseling, which really also isn't all that surprising. And then the diversity decreases over time. So not a lot of them are saprobes, which are fungi that feed on dead tissue. So very few of them would be classified that way. So it makes sense that we're not finding them in dead tissue at senescence. But what was really, I think, really cool and probably really cool for your listeners to know is that Fusarium graminearum, right, associated with gibberella ear rot and stalk rot, we found was ubiquitous, so always present across plant development and between management styles in leaf tissue, starting from early vegetative stages, so like around V5, all the way to R5 and senescence. So the question being, what does this mean for things like mycotoxin contamination? We don't know. What does this mean for the context of disease symptoms and disease severity? We really don't know. This is a fairly new result, and is really sort of the next, I think, direction this work can take, and sort of exploring what this actually means for growers.
And so maybe this is kind of what you're saying, and I'm just, what triggers that fusarium to go from something actively living on a leaf and being non-detrimental to the plant to now showing up as head scab or stalk rot or ear rot? Or is that the question, or do we know that answer?
We don't know. We don't, I mean, very broadly, so like in plant pathology, we have the disease triangle, which is a way that we characterize disease outcomes, right? So you have a virulent pathogen, a susceptible host, an optimal environment. And so very broadly, you could say that environment optimal for disease could be a trigger, right? That really matters in the context of management. But we really don't know what that transition, like where that happens, why that happens, if it's genetic, if there are specific, individuals that are more virulent than others. We don't like really know what that transition looks like. And there are people exploring this in other Fusarium, like Fusarium solani, in like tomato and things like that, or crops like that. But we really don't know yet.
And so my last one about that, like kind of looking at this, how is this applicable to the farming community, and where does this go? If I'm dealing with someone that's got wheat or they've got corn and they have a lot of gibberella, our advice is rotate out of that grass crop, right? Go into a cover crop, go into beans, go into something else, get that residue broken down, try and get that out of there. But if we're saying it exists on the leaves of the beans or all of the other cover crops potentially, or maybe it's not potential, maybe it's going to happen, is that still, are we looking at like, are we ever truly going to get rid of this? Probably not, right? And so how far do we stretch that to make this effective? Or maybe that makes sense, maybe it doesn't. But how do we use our rotations to help control this if it's always present?
So I think before I would have 100% said, again, I'm not an extension agent. My research, can be applied, but I can't make any recommendations. But before before sort of finding this result and having conversations, uh, I would have said that the rotations we're using right now would be perfect, right? You're rotating out with a non-grass host, or a non-grass plant crop, and so you're likely going to sort of reduce inoculum in those fields. Um, I honestly don't know. I don't have any like major suggestions on this. And the reason being is that from what I've heard from other extension folks is that they're finding graminearum in soy. We have other grasses that grow either as cover crops like triticale or as weeds. And so it's really the idea being that this pathogen is always going to be present. And so maybe thinking about other angles of management. I know a lot of people are thinking about like reducing weeds, and I think that that's always a great focus. I know like one of our major agrochemicals that we use in corn is really herbicides. Um, but my thought would be reducing weeds and then thinking about other areas of management and mitigation.
Yeah, I think that's a great point. And how do we learn from some of this? And I've spent a little bit of time thinking about this too, because I've kind of seen some similar patterns. And one of the things that when I find it interesting that the grasses. So when we're talking about triticale oat, are they drivers for an inoculant buildup? Oftentimes, I think all a living green bridge is just always kind of gonna allow for some inoculum buildup. And these pathogens, some of the fusarium pathogens, you've used the word ubiquitous a lot. And I think for some of those cases, it doesn't really matter. Now, I will say in the literature, there is evidence that when you have full heads, so in a case of cover crop, right, you're terminating it before you have head development. Now,
Yes.
if you have a really scabby heads in your field, yeah, you're going to have higher levels of local inoculum. So I'd say that I'd add that when we're talking about a cover crop, maybe it doesn't matter as much for fusarium. Maybe some other root pathogens it does, but I think when you actually get to having wheat that heads, a dense mulch of that, that's where you get into problems with having some inoculum buildup for those ear rots. But yeah. Well, I think that's all very interesting. I think Justin seems like he has a lot more questions that he even wants to ask.
You know what? It's the thing about these podcasts is I spend 20, 30 minutes with someone, then I have the rest of my day to kind of ruminate on this and develop this like, well, what about this and what about that? So, over the next 3 days, I'll just be hitting Ryan saying like, man, what about this one, right? What do you think? So, uh, I think this is amazing stuff. And you mentioned that you're not an extension agent and you don't make recommendations. Like I get that part, but even within extension, we get really detached from this kind of stuff too. So it's always fascinating for me to see what's actually going on in the science that the university is doing to help. 5 years from now, I may be giving a talk on the importance of this within our cropping rotation, right? And you just don't know what's happening and where these things come from. So it's been really amazing to listen. Thank you.
Thank you so much.
All right, next on we have Allie Rice, and Allie is a master's student in Dr. Carolyn Lowry's lab. And I'll let her introduce herself and some of the work that she's doing and her lab's doing.
Hi, everyone. My name is Allie Rice. And as Ryan said, I am advised under Carolyn Lowry as a master's student. And a lot of my work focuses on studying the seed bank community, the soil seed bank community, and what's going on under there. So, the perennial forage section of the project is what my research is based upon. And some of the baseline data that we've taken, for example, some of the things we're looking at is what weed seeds are in the soil seed bank, and what does that look like compared to above ground weed species? And are these perennial forage treatments making any difference, positive or negative, in the weed seed bank community?
Okay,
So
awesome. So
I work a lot with seeds.
gotcha. So and
Mhm.
specifically you're talking about weed seeds. And
Mm-hmm.
so my question, so I introduced the project, so our audience has a good idea of what's going on. And when I say the project, the cover crop cocktails plot, right? The
Mhm.
rotations that are going on are normal corn, soy, wheat cover crop rotation. And you kind
Mhm.
of said that you're looking at this new perennial addition to it. So can you talk a little bit
Mhm.
about what that perennial system addition is and why they added this and kind of give us a little bit more context on that?
Yeah, they've been studying in the cover crop cocktails field for about a decade now. And so we're starting to see how the weed community is acting in those annual rotations. But now we are trying to kind of push perennial forages because of the soil retention benefits. There are known weed suppression benefits. So we know what the annual rotations are looking like. We want to introduce this new um, perennial forage idea because a lot of organic farmers are experimenting with it, and it's kind of based on the questions that they ask. They would like to know, do these perennial forage treatments, how do they affect the soil, the nutrients and the retention? How do they affect the weed community? Because there are a lot of positives to having perennial forage uh, crop in the field. You have that long-term cover, weed suppression, soil retention. So we just kind of wanted to look more into that. And sometimes organic farmers have a hard time finding a market for their perennial forages. So we want to see, well, if there's weed benefits and weed suppression benefits, that might be another positive for organic farmers to introduce more perennial forages into their rotations. So we just wanted to go off the idea of what more can we do with these forages? And we already have this annual rotation project. Let's compare annual rotation to the perennial.
So when you're cataloging or kind of how do I say this? Maybe broadly speaking, what are the weeds that you're really keyed into as far as seeds and the above ground populations? And I say that with the preface of like I work in annuals and I work in perennials, and often they're very different. Right, the spectrum of weeds that we have to deal with in a hayfield versus a cornfield, hopefully, if we're managing our hay well, is not the same thing. So, how do you decide what you're really focused on?
Well, the main two species in my thesis are surrounded by giant foxtail and Canada thistle. And those were kind of decided to be some of the two main study species because they're very prevalent in our research field. And particularly with giant foxtail, we see very vast differences in the populations among the plots. So we already have like an interesting giant foxtail and Canada thistle community going on that we decided to investigate how the perennial forages may or may not affect it. And mainly Canada thistle because it's such a problem for organic farmers. As far as annual species, you have it right on. How do you tackle so many different diverse species, perennial and annual? And so we decide what annual species we're studying based on what's there. So, and how much of it. So a lot of what we focus on is based on what we've seen. So I focus a lot on corn speedwell, lamb's quarter, pigweed. Pigweed not as much, surprisingly, but a lot of lamb's quarter, scarlet pimpernel, common chickweed. There's about 10 to 12 main species that are popping up in very large numbers in the seed bank. So we kind of let the research field tell us what species we're going to study based on what's there. But also, again, the Canada thistle, farmers really need some research going on to how to handle that. So that's one of the reasons why we picked the thistle as a main perennial to study. Yeah.
So are you finding that, how are you finding that I guess you said that this perennial system that you've added is in its first year or one
Second year.
second year. Okay.
Yeah,
So are you
basically.
finding already after you kind of take these annual fields into production of a perennial, um, have you found that this changes the seed bank or helps you manage some of those other perennial problems as well?
So as far as the soil seed bank, we'll get a better idea of how the perennial forage treatment is affecting it when we take our data again at the end of my master's program. So in 2 years, we will know the huge, the hopefully huge differences that these perennial treatments are making. But right now, even just having the baseline data, we can go into the field and visually see how these species are reacting. For example, we see that in the monoculture forage chicory plots, there is a lot of weed suppression, even though there's those little gaps in between, like the basal structure of the plant. We're finding that there's not a lot of weed species popping up there. So when we go and take data again after these perennial treatments have been in the ground for a while, hopefully we can confirm that that chicory was suppressing those weeds. So we have seen a lot of visual, interesting notes in the field about weed suppression. So yeah, one of the biggest things that I have seen is that this forage chicory is suppressing annual and perennial weeds. I don't think it's alleliopathic. I think it's the structure of the chicory.
As in the above ground structure leafing out enough to call shading to stop some of these smaller things, or how do you explain that with something like Canada thistle that's typically coming back from, rootstock as much as it is seed. That's an interesting one.
Right. And I think that's as we keep growing these treatments, because again, I'm really mainly seeing that in the treatment that is 1 year old. So planted last year and it had a full year of growth. And I'm seeing in that, again, it is hard to explain because plants act in such weird ways. There's so many different conditions. But I think when we go to take that data at basically the end of my master's, that hopefully we can see why. Because like you said, that thistle should be shooting right back up. Its roots are in the ground. So right now we don't really have too many answers as to why.
But that's okay.
Yeah.
That's part of the journey, right? We'll just have to set our clocks to when you finish your master so that we can come back and do this again. And then you're going to just throw out all the answers for us.
And I wish, that's, as you guys know, that's kind of the hard part of being a scientist is we want to give these answers, but it does take time. But I am very excited to see how these forages are going to react because like I said, I've seen visual differences. And one thing we're kind of doing to ensure that we are documenting the populations properly is we are using a satellite GPS unit. And in each plot, we marked a no thistle and high thistle point. And so each time we do our biomass harvest, like 4 times a year, every year, we go to that exact part of the Canada thistle population in that plot and see exactly how it's doing. So I think having, that satellite GPS is really going to help us give some nice accurate data as well.
Well, that's interesting. I know variation is always a problem with some of these studies, and so that's a cool way to kind of cut down on that, where you're selecting a certain area and watching how it changes. So, you brought up forage chicory, and maybe I should have asked this earlier on because I was just thinking, okay, your perennial system's probably an alfalfa, grass. But then you said forage chicory, and I don't know anything about it. So, what are the treatments that are being tested? Is it just forage chicory? Do you have alfalfa next to it to compare? Can you tell me a little bit more about what other treatments are out there and why you guys are looking at forage chicory, where the interest is there? Because I'm not familiar with it.
Yeah.
Yeah, we're actually studying a lot of different, we're studying 6 different species, perennial forage species, and we have monocultures of each, and then we have like a 2 species mix, 3 species, 4 species, 6 species mix. So we have different mixes of each type of forage. And so we study alfalfa, orchard grass, small burnet, bird's foot trefoil, forage chicory, and tall fescue. And a lot of those are, farmers typically, you think alfalfa and orchard grass, but yeah, some of especially the forage chicory is interesting. A lot of people include that when they, as far as I know, they include that in their rotation when they're using their forages for cattle feed. So I think that's kind of an animal draw, a livestock draw, I believe. But I think one of the most interesting species would be the small burnet. That is a forb that comes from Illinois. And I've heard that it's hard to find forbs, good perennial forage, forbs to include in a rotation. So including that small burnet, there's these species that people generally don't experiment with, and we're kind of doing the trial and error for them. So there's 6 different species we study.
So when you're, and maybe this was something that happened before you got there, when you're seeding this and you're talking about this being a monoculture, I guess for us in pastures, or hey, like that's as we transition it, how are we establishing a monoculture? Are we going in and like tilling or chemically burning down? You're an organic system, so you're not chemically
Yeah.
burning down. But how are you establishing this monoculture with all the other diverse plant species that would be there initially?
I think a lot of the monoculture, like, because we have a monoculture of each species, I think the purposes of that is to see how those species react alone. Not necessarily that, unless, there's some super benefits, not necessarily that we're recommending that farmers plant those monocultures, but just so we get an idea of how they're reacting, what's the forage quality on them, and the type of harvest we're getting. Is a certain monoculture suppressing weeds? Then if it is, then we might look into, okay, how can we help farmers establish that monoculture? Because yeah, that would be quite difficult. So I think that's a little bit more for our purposes right now. But those 2, 3, and 6 species mix, I feel like would be a great tool
And,
as
and
well. That
in for the context of, I'm a little slow. It takes me a while to put all this in the right
No, it's
order.
okay.
So if we're thinking about this, what we would be looking at is potentially like an organic corn, soy, small grain type rotation that we might have 2 or 3, 4, whatever years of an annual, and then we're putting this into this rest period in a perennial with some type of mix for 2, 3, 5 unknown amount of years, and then we switch back to our annual cropping system. Is that right? That's kind of the hope with this, or are we thinking of these as more like a fall cover crop? And in that case, the perennial part doesn't. We wouldn't need to go that route.
right. Yeah, we're kind of looking at yeah, introducing that into a part of the phase, because, and that's the hard part, is because who wants to let their field sit, right? And that's why we're saying, well, there's all these benefits, you know um and you can, in some ways make money off the perennial forages. But yes.
And I get that, right? 10 years ago, 15 years ago, 2050, when there were more dairies, the row cropping with a perennial aspect made a lot more sense, right? From a nutrient management side, from a weed management side, we would do some row cropping, then we would go into some type of forage for 4 or 5 years, whatever that was. We could get some
Yes.
fertility back. We could get some soil
Hmm.
health. We could do now as and this is my perspective, maybe that's not right, we're really starting to separate our row cropping and agricultural systems. And so that's the
Yeah.
hard push that I have with even my farmers that are non-organic when I say like, let's put in some sort of small grain cover. Well, what are we going to do with that, right? We don't want to use triticale because that's an animal thing. Or if we talk about going into something like that, there's a lot of pushback because you have to have the mouths to feed it to make it make sense.
Yeah, so you definitely get the struggle of, but sometimes, if it's not that cash crop, it's hard to see, the benefits. So yeah, we're looking at how can these, and that's the thing, forages some people say that you got to have it for up to 4 years for it to be, super beneficial. So that's why we're kind of looking at what are the benefits of having that long-term rotation, because in this project we're looking at up to 4 years. So the first forage that we planted, that will be 4 years old when it's terminated. So we can see that long-term effect it may have. So I don't know if I kind of strayed off from your question, but yeah, that's the idea, is that it would be the annual rotation and then a couple years of perennial forage.
I would love to see more forages in the landscape. So keep up the good work, and we just have to convince people to do it now. We just gotta get buy-in.
Yeah.
The last thing I will say is I appreciate you saying Canada thistle instead of Canadian thistle. If I rank my pet peeves as an agronomist, that's right up there in that. So I appreciate you understanding what you're working with and using the correct pronunciation. Thank you.
That's great, of course. Yeah. Yeah, I'm really glad I could come on today and share, because I agree, pro forage, pro perennial forage. And that's why we're really excited to look at these species that, organic farmers sometimes aren't, it's kind of new to them, and that a lot of people haven't been working with, like the small burnet and, forage chicory. So I love the idea that we're trying to get some new species out there too as well to convince people.
That's right, and I can't wait to see what you find on that weed suppression end, because I think one of the things in this cover crop cocktails plot work over the last 13 years we've realized is that there's a lot of weed populations that build up even when you have cover crops, when you have living green tissue, a little bit of
Yep.
tillage, like it's really hard. So anything for the organic farmer that can help with weed suppression, and then if we can find a good way to encourage perennials, man,
Mhm.
what an awesome system that would be. So I'm
Yeah.
happy to hear that you guys have included that in the project, and that you're looking specifically at the weed management benefits, because that's a huge issue. So thanks again, Allie, for coming on. We appreciate it.
Yes. Thank you guys so much.
And Allie, we're going to attach a link to the USDA Small Burnet Management Guide, because I think that might be a plant a lot
Ooh,
of people aren't familiar with. I learned about it last week when Allie presented to a group of us, and spent a lot of time Googling after that, because we had no idea,
Nice.
right? And it's a lot of forage people. So we're going to throw that in there for anyone that's interested in small burnet. Take a look.
All right, and to our listeners, thank you for sticking with us through those two interviews, and we'll catch you on the next episode of the Agronomy Highlights podcast.